Hello my dear students, I hope all of you are doing very well. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute. So, we are continuing our learning in quality assurance. So, my dear students, since last many of the lectures I have told you why quality is important and how you can ensure the quality in our pharmaceutical products. So, we will continue from today. I am going to start a new unit, unit 3, and this is the 15th lecture of our this subject. Today, I am going to tell you that how you can do the quality control test for various containers and other kinds of that is substances like rubber closures. So, let me start. So, these are the things that we are going to discuss. I will tell you introduction to quality assurance in packing, then I will tell you the quality control test for glass container, then chemical resistant test, hydraulic resistant test, arsenic test, thermal shock test, and including internal bursting pressure test and other quality test for closures. So, let me take to you just very basics. I think already you have seen this slide in my previous lectures, where I have mentioned you how the entire organization is actually that is a organization structure is actually working. So, all of you are aware that quality control manager will be there and quality assurance manager will be there. There are lots of people, lots of chemists who will be working under them. Okay. What about the production activity will be happening under production manager? The substances or the tablet, capsule, syrup, whatever the things they will produce, that thing will be analyzed by QC department under QC manager. The QC chemist will analyze and QA chemist will assist, they will supervise everything. Okay. I also told you that what is the workflow of a typical pharma industry. You have seen that in warehouse all the raw materials will come, and then QC chemist and QA chemist, they will be associated with analyzing its that is its quality. The raw materials whatever is coming, its quality will be analyzed by QC and QA chemist. QA chemist will collect the sample and they will give the sample to QC chemist, they will analyze in the lab, and after that, after analysis, the QC chemist they will give the certificate that yes, this is perfect, and if it is not perfect, if it is not falling under the limit, then they will tell that no, it is failed, it is actually not approved. At that scenario, after getting the certificate from QC chemist, QA chemist will assist to bring this thing into our production area, and here the production activity will happen. Now, what are the things I am trying to tell you regarding containers, closures, regarding packing? When in warehouse, I have told you raw material, packing material is also included within raw material. Okay. Now, so packing material is also included in raw material. So, the QC chemist will analyze the packing material, they will check whether it is perfect or not. There are lots of tests, I will tell you in detail about all the various tests. Now, let me tell you some basics things, why packing is important and what is the basics understanding of packing. So, packaging of pharmaceuticals products gives an adequate degree of protection, minimizes the loss of constituents, and should not be interact physically or chemically with the product. So, you need to do packing, either primary packing, secondary packing, tertiary packing, you need to do all kinds of packing. There are some products which are actually we go only for primary packing and secondary packing, and there are some products we are going for the tertiary packing also. I will tell you more what is tertiary packing, do not worry. Now, packing is a process by which pharmaceuticals are actually suitably packed, so that they should retain in their therapeutic effectiveness from the time of packing till they consume. Means after production you have to do packing, and after packing it will be go to the market, patient will buy this and they will consume that thing. Till the time the drug will be under this packet. If I tell you more, if I tell a definition, it is defined as the art and science which involves preparing the articles for transport, storage, display and use. Transport, storage, display and use. Okay. Pharmaceutical packaging is the means of providing protection, presentation, identification, information, convenience to encourage the compliance with the course of study. You see, I have used a lots of words in a single line. So, what I am actually trying to tell you? I am trying to tell you that transport, there are chances during transport, if your substance is highly friable, there might be some issue with its that is the actual form. Storage, for storage, if you are trying to keep your substance in a very low temperature, the storage will come to the picture. Display, if you do proper packing, after packing in display you can write the name of the product, all the labeling things, expiry, manufacturer, batch nom, everything will be there. If I tell you more, protection already you are aware, presentation again it is related to our this thing called display, identification is also important, and information, there is something called PIS. Whatever the information are actually available in PIS, some information we actually also try to provide in packaging, in the labeling, okay, and convenience to the patient. So, that is why it is important, and this is a very simple definition for packaging. Any material which used for the packaging of the product for sale and transportation said to be packaging material. Okay. What is the definition of packaging material? Any material which is used for packing of the product for sale and transportation will be said to be packaging material, any material. Okay. It might be your primary, secondary, tertiary or any substance which is associated to make the packing material. If I tell you more, commonly whatever the packaging material used, these are container, closures and carton. Containers, closures and carton, primary, secondary, tertiary. I will come to in my next slide about all the details. Container, whatever the, now what is container, sir? Containers mostly made of your glass, plastics, metals or paper. The material for closures are include the cork, glass or plastic. Okay. See and see. Now, let me take to you in this slide. Here I am actually trying to tell you because lots of times, several times I have used this word primary packing, secondary packing, tertiary packing. So, now I like to tell you about the primary packing, secondary packing, tertiary packing. See, what is this? What is this? Many of you might be telling me that sir, this is tablet, this is tablet, this is tablet. Hai na? Now, whatever I am trying to tell you since the beginning of my lecture, we have to protect our this tablet. We have to tell the people that what is the name of this tablet, and we have to protect the tablet in such a manner that during transportation its friability should not be changed. That is why first we are doing primary packing. Primary packing, you have strip, you are putting your tablets here, primary packing. Then you are doing another packing called secondary packing in such kind of boxes. It contains many units of primary packaging. Lots of your this primary packaging material can be primary packed substances can be added here, and then carton called tertiary. I hope now it is very much clear to you all after observing this picture about the primary, secondary and tertiary. I have taken this picture from internet, so copyright is goes to them. I am using this only for educational purposes. Now, in primary packing already you have seen that the substance which is closely come with the contact of our drug substances. If you have prepared tablet, primary packing material will be completely closed with our substance. So, for example, these are the more detailed picture of primary packing. It contains direct contact with product, bottles, vials, ampoules, tin, blister, package, strip, etc., etc. What is secondary? Already after watching my that slide, you are slightly aware. Let me take you to more. So, in medicine shop, whatever you are observing, in medicine shop, maximum of our these products are actually available in the form of secondary packing. You see, this is the outside in the primary packing. Perhaps used to group primary package together and cover it like this. If you open all those things, you will be getting multiple primary packaging material. And tertiary is the these things carton. Sir, why carton is required? Because you see in a country. For example, I have worked in a pharmaceutical company which is actually used to that means export pharmaceutical products to African countries. So, when we used to export our products in African countries, we used to export for a large amount of substances. Large means at least 1 million, 2 million, 3 millions of the tablets. So, if you send a large amount of your tablet, capsule, syrup, so you need to do tertiary packing, where multiple primary, secondary substances will be put it here. Now, how you will evaluate and what should be the ideal requirement for a good package? They should be able to hold the product within loss on account of leakage, spoilage and permeation. Leakage, spoilage and permeation, very important. They should afford protect against environment condition, like light, air, moisture during the storage. Light, air and moisture. I can remember I was reading one story when I have joined our pharma industry. My boss actually given me a book where all the failures was written, where all the packing failures were written. There I was actually reading that some pharma company, it is actually from Asia, not from India, in Asia. I do not want to take the name of the country. That country sent, that is exported some product in South American countries. What happened? Their packing material was not good. Due to that, it was the time of I think rainy season, when it has reached in South American countries, due to the high humidity in those area, moisture entered into the product, and due to that it has lost its property. It is a failure. That is why when you do packaging, you should try to maintain to protect against environmental condition like air, moisture, etc., and they should not have any permeability for glasses, and they should not possess, they should possess sufficient strength with withstand shocks, handling and transportation. See, in our India, we all of you are actually aware that the status of our transportation system. Our roads are not very good if compared to the developed countries. So, we all of you are aware that what will happen when the products will be go with auto and truck and other things. So, if our product is highly friable, then it will be not in its actual condition because of the poor transportation, poor condition of the roads. Not even our country, in African countries also situation is same. So, that is why it is very much important your packaging material should possess sufficient strength to withstand shocks, handling and transportation, etc., etc. I hope you are getting my points and you are also enjoying the learning. If I tell you more, there are few other requirements. What are this? They should facilitate the sufficient, efficient, safe and convenient use of the content, and also the material, the material must not interact with the content. This is very important. The material must not interact with the content. All of you have studied that drug interaction, drug food interaction. Now, another thing you will have to study about drug packaging material interaction. You all earlier you have also studied the drug excipient interaction E. Okay. So, you also have to understand that drug packaging material interaction. In very less amount of cases, it is observed that drug is actually coming in contact with your packaging material and then it is degraded. So, usually it is not happening until and unless you use the low grade packaging material. If you use low grade packaging material, on those scenario only such kind of problems can arise. So, you will have to take care of these things. Then the containers should afford protection from the modules, various bacteria, etc., means I am trying to tell you about the microbes. Microbes can be harmful. your packaging material or your container should avoid the protection. There are lots of microbes, they can pass lots of membranes. If they pass, obviously they will interact with your drug and it will create a problem. The cost of material should be as low as possible without compromising the quality, low cost. But in every cases we cannot that is a afford that is a high cost material, but to if you want to make a good product, if you want to make a quality product, you have to understand and you have to design based on the cost. Maximum of the pharma companies, they are try to reduce the cost by compromising with the packaging material, but I will suggest that this is a wrong practice. Do not go with these kinds of wrong practices, because ultimately you have done tremendous hard work. Your production people are working, your QC, QA, DRA, all the peoples have worked. After that you got to your this ultimate formulation. Just using a low grade packing material, if it may degrade your ultimate formulation, then there are no use. Ultimately, any of the drug control agency will catch your this substandard drug and they will ban you. So, do not go with these things. They should facilitate easy identification and also they should afford protection from mold sources. I will mention this point, and the container should be should not absorb any material. Now, what are the tests are we going to do? You have told a lots of thing about our this packaging. Now, how we shall able to check that yes, our packaging material whatever we have bought, it is correct. What are the ideal quality control test? So, there are lots of tests, but most the commonly used tests are these things: chemical resistant test. There are two types of test: powder glass test or water attack test. Then hydraulic resistant test, arsenic test, thermal shock test. Also you can do internal bursting pressure test. Other tests are also available. I will tell you about the other tests in my subsequent slides. Now, first is the chemical resistant test, under which we have powered glass test. So, powered glass test, how you will do this test? There are two steps to do this test. What are the steps? First, you have to, in step 1, you have to do preparation of the glass specimen. Preparation of the glass specimen. How you will do this? Few containers are rinsed thoroughly with purified water and dried with steam of clean water, clean air. Grind the container in a mortar to fine powder and pass through sieve number 2050. In step 2, washing of the specimen. 10 gram of the above specimen will be taken into 250 ml of conical flasks and then wash it with 30 ml of acetone. Repeat the washing and descent the acetone and the dried, after which it will be used for the 48 hour. This is the step. After that you can get the some results. Now, if I tell you the entire procedure, if I tell you more, you can take the 10 gram of the sample with the 50 ml of the high purity water in the 250 ml of the flask, place it in an autoclave for at 121 degree centigrade or plus minus 2 for 30 minutes. Then you have to cool it under running water, then descent whatever you have got in our previous work. In step 2, descent the solution into another flask, wash again for 15 minutes with high purity of water and again descent, titrate immediately with 0.0, that is normal sulfuric acid HCl, using methyl red as an indicator, and record the volume. Now, for different substances, for different packaging material, after doing titration, there will be limit. There will be a limit. If your product is correct, if your the packaging material is correct, at that scenario it will be within the limit. So, ultimately titration will be give you the final result. Yeah. Now, so what kind of areas or what kind of container we can use? So, type 1, type 2, type 3, all of you are aware that these are the 3 different types of container. Now, whatever the volume we have used here, that is sulfuric acid, its normality, it can be varies. Okay. Based on that, for type 1 it would be 1 volume, then for type 2 it will be 8.5, type 3 it will be 15. Similarly, you can also do water attack test. This test used only with the container that has been exposed to the sulfur dioxide fumes after controlling the humidity condition. Such test neutralizes the surface of alkali. This principle involves the water attack test is determine the whether the alkali leached from the surface of the container. Okay. So, these are the various types of your that is glass material and what kind of test you may that is follow. This is the entire step. I am not going to discuss in detail because of the shortage of time. Then again I have mentioned you another test you can do called hydraulic resistant test. This is very simple. To do this test, you have to rinse each container and fill with the water, then heat with this temperature 100 degree and rinse to the 121 degree centigrade calculate at autoclave, then remove from the autoclave and combine in the liquid, then titrate. See, again and again ultimately titration is coming to the picture. If it is after titration, you will note the volume. If it is within the range then or limit, then it is correct, otherwise you will have to that is certify that it is not correct. These are the details you can read in your free time by pausing the video. And another test you can do the arsenic test. You can wash the inner and outer surface of the container with fresh distilled water and prepare the test solution same as the hydraulic test, and then in the third step 10 ml of the test of solution plus 10 ml of the HNO3 to be dried at 130 degree centigrade in oven, then cool, then add hydrogen this molybdenum reagents, then heat under water bath and cool and absorbent the you may see the absorbance under 40 degree nanometer. Thermal shock test is also used. What we are doing here, we may place sample container in uprising position in tray and immerse the tray in hot water and given some time, and then we transfer the container in cold water bath and temperature should be controlled, and then we examine that before and after test. We examine that before and after the test, means before if there is any crack is present, then it is we can assume something, but if after that is giving the stress, after giving the temperature, if there is changes, then obviously there is some issue. You can do other test like internal bursting procedure test, and you can do also leak test. I will discuss about leak test, because many of the pharmaceutical products they actually get encountered with moisture and other thing just because of the leakage. This is very simple, this process is very simple. If you are in lab, a simple lab, you can try this kind of test. When I was in pharma industry, I was also the in charge of IPQC in process quality control test. So, whatever the substances, whatever the packaging things are they are doing our worker. So, I was supposed to go to their working area, take some sample randomly, take some sample means packed sample drug with packet. Then I used to come to my lab, that is IPQC lab, and I used to check. So, your drug filled container will be placed in a container filled colored solution. Due to the addition of the dye, it will be give you some color, which is high pressure compressed compared to the pressure inside and the glass container. So, that color solution enter in the container. If any crack is present or breakage is present, this if when you open your this tablet capsules other things, it will be colored, very simple. There are other tests are also available for closures. You can do sterility test, fragmentation test, then you can do self solubility test, pH for aqueous extract, you can do light absorption test, then reducing the substance, residue evaporation and permeability test. So, today we have actually learned that the introduction. We also have seen the quality control test for various glass container, the chemical resistant test, hydraulic test, arsenic test, thermal shock test, internal bursting pressure test, and also quality control test for closures. I hope all of you have enjoyed this lecture. In next lecture, I am going to tell you more about our understanding for the secondary material. So, thank you all of you, my dear students. we may meet in our next lecture. I hope you are getting all of my points. You are also learning, your understanding with this subject is increasing a lot, and my lectures will help you in your today's examination, your B Pharm course, and also will help you during your industry time. Thank you all of you, all the best.